Good morning, everyone. Dear organizing committee, thank you very much for the invitation and the introduction. As you have just said, I am Julia Sidorova, and I work at a consortium of hospitals doing bioinformatics clinical analysis. And the topic of my talk today will be deep learning-based survival analysis of omics and clinicopathological data. This is a preamble to my talk. You can see Picasso's Don Quixote drawing, and it's not because that I'm giving a talk from Spain, it's because of the essence of the talk, the meaning of which I'm sure will be clear by the end of the talk. Currently, in the modern literary analysis, there's a fancy interpretation of Cervantes' Don Quixote, that this is the same person, just it's two sides of the person. Sancho Panza is someone very careful and can't even look at what Don Quixote is doing, and Don Quixote is audacious and a little bit crazy. The agenda of the talk today will be, first we'll define the context and typical problems of clinical bioinformatics. Then we will revisit classical survival analysis and explain the need of deep-based survival analysis, the need of the emergence of this paradigm. Then we'll touch these situations where conclusions from computer science do not hold for other subfields and fields where the analysis especially is mission critical. And then finally we'll talk about the artificial intelligence constraint called the brain. The context, clinical bioinformatics analysis. First, the type of data. The type of data here is definitely not image. If it would be image, everything would be different, because then the conclusions might change. Why? Because for image, definitely convolutional neural networks are a very suitable tool, and it takes a lot of argument and possible to say that something else is more optimal, and the conclusion can be different. Conclusion from my conclusion of this talk. So image is not included. Even when we talk about radiomics, I'm talking about radiomics, the old feature extraction system, which image is an input and the output is the comma-separated numerical data. Clinicopathological variables are, the so-called clinicopathological variables are gene counts and some key attributes. The key attributes are some features that are known to be highly relevant for the application. It can be sex, it can be age, it can be something else very specific to the disease and pathology. For example, as you see in the purple font, 00100, something else, numbers, then 30 years, male, some specific attributes, and then in the bold font, the variable of the interest, the endpoint of the response variable. This type of data is often fat, which I mean that is characterized by the condition that P is much greater than N. P is the number of attributes and N is the number of data points in the data set. It also is informally referred to as curse of dimensionality. On top of it, data can be sparse, Quite often it is sparse, a lot of zeros. It can present some drift. Drift is context drift. For example, the typical example in computer science would be summer and then winter, and then the patterns change. And something else which I cannot see because I can see my video. All right, what I meant to say, possibly noisy or not with missing values. Basically, what is meant to say by this, that this is numerical and data with complications. Most typical analysis a bioinformatics platform confronts with is typically RNA sequencing, which is, for those outside of the field, finding up and down regulated genes. Say there's a pathology, disease, something, condition, and then sequence the genome and see that for an individual with that, some genes are down-regulated and other genes are present with a greater count, so would be up-regulated. Second typical task is analysis of group of genes. So now what was done at step 1 would be group by function or based on computational properties. So that's the same, but some grouping of one sort or another. Third thing, survival analysis. You'll see quite a lot of it in the coming slides. Then classification and regression. Then recently very popular single cell. So that would be not at the level of population as above, but at the level of the cell, not at the level of the patient level, Recently in the Nature, I read that engineers and computer scientists, bioinformaticians from the Silicon Valley say they will automate bioinformatics analysis, and even with a voice interface. Leaving alone the voice interface, which looks a rather trivial task, all right, that's okay. Automate bioinformatics analysis, would that be plausible? Currently, I don't think so, because quite often clinicians contrast my analysis with what ChatGPT tells them that would be an appropriate analysis. Well, ChatGPT is invariably wrong when we diverge. And you see, I'm playing with the font here. Green, a promise from the Silicon Valley. And the purple, that's it's crossed out, so that's incorrect. So this is where Sancho Panza and Don Quixote is coming. Don Quixote gives algorithms, gives ways under promise, and then there's a clinical statistician or whatever, a practitioner or an analyst, as this profession is sometimes referred to. And then it turns out that it's a very careful person who is not so much, or not in the first place looking for functionality, but someone who is looking for a statistical guarantee, for inference. As I said in at the first slide, I dedicate my work, my days, to mission-critical data analysis for a network of research hospitals. So here, given the context, things as p-values and false discovery rate and other inference parameters are highly important. Not just say, oh, it's a predictive model with a great success rate. No, that's not enough. So you see purple, the word inference again. I would say that our field of practitioners, of clinical practitioner, of data analysts, is completely different, disjoint in terms of journals and notions and success metrics from computer science algorithms, in terms of language, in terms of quite a lot of things. What would be the journals one would publish? I've taken recently the cuts of the recent journals. That would be Nature Communication, Hepatology, Journal of Neuroinflammation, Clinical and Translational Gastroenterology. So these are not the journals where an algorithm would be published. Organizing a special issue, I was warned by the editor-in-chief, asking, please do not include the word algorithm in order not to attract the public that is definitely outside of the scope. So you see again the interplay of the fonts, where the algorithm world is comes into and a world of the inferential statistics. I remember when I was a student, we were laughing at an old joke that there's a surgeon, a person who can do everything, and a pathologist who is dissecting the dead body and identifying the cause of death. So that person knows anything. Surgeon knows nothing but can do anything, and pathologist knows everything but can't do anything. I think it also can be a quite suitable metaphor of the state of affairs. Very needed, very new, indeed very sought after possibilities from computer science, green font. But they must be translated and studied in the context and from the prisma of the objectives of the application. Coming to a conclusion, a statement that without inferential parts, conclusions can be wrong or optimistic, as they optimistically write in computer science books. It makes me remember Noam Chomsky with his famous article that green ideas are fiercely asleep. That was the famous title of his article when the generative linguistics was starting. All right, so green. And just having given a talk in Salamanca, the symbol of the city is a skull, death. So conclusions will not hold at all. And the green frog, a symbol of a sin, is sitting on the skull, saying that if one does not follow the proper protocol, then most likely death. Or optimistic conclusions. Second part of my talk is dedicated to survival analysis, which is the following thing. Given the patient's attributes, so say gene count, sex, age, something else relevant to the condition or disease under examination, there's also the response variable is time t, which means survive past t. Say patient with this diagnosis, treated with this treatment, survived 3 years after the start of the outbreak of the disease. From 1955 to 1975, survival analysis was developed as a discipline and adapted for application in biomedical studies. It is very much like a regression problem. It perfectly would be regression, if not for one nasty mathematical complication. This mathematical complication is this: data is censored. That is survived past the end of the study, but survival time is unknown, for example. So there would be a lot of lines in the database file where there are attributes, but the survival is unknown, possibly because the person left the study, or possibly because the person survived, fortunately, by the end of the study, so was still alive when the study ended. Very well. It would be possible to ignore the lines, yet that's not what one wants. And there were different approaches and different algorithms, but finally what became the gold standard of the clinical statistics is the survival analysis. It's a set of ingenious algorithms where smart statisticians, including Cox, found a way around. So we say one should not use regression, and also one cannot use a two-sample test to test whether two groups are identical or two groups are not identical. By 1975, all the problems were solved, and as I've said, it has become the gold standard in clinical statistics. So let's briefly revisit, or hear about here for the first time, possibly, what are the main problems, what are the main three problems in the statistical analysis. First, it's a Kaplan-Meier estimate. That's a graphical comparison. Say one has two groups, one was treated with chemotherapy only, and the other one with chemotherapy and radiation. And graphically, given that Y is survival, so that's the percent of the population still alive at the first time moment, everyone is alive, and then people unfortunately are dying, and the percent given days go on, and fewer patients remain alive. So all right, two groups, and some are doing better, seemingly, maybe not. So here's where the inference comes. So are they really doing better, or it's just a graphical variance without any significance behind? The graphics, I guess, comes from Efron clinical data analysis. Anyway, someone very central in the field. I guess that was Efron inference at the computer age, statistical inference at the computer age. 2022 book, wonderful book. Second, I said that a student test is not good enough. So again, we've seen that two groups, one is seemingly graphically at a graphical representation doing better, and a log rank test is a non-parametric analog of a two-sample test for censored data. So that's for our data. I remember I mentioned what a nasty mathematical complication it has. So it gives an answer to the question, is group A really doing better than B, or that's just random variability? Right. The last blow on the difficulty of the problem came when the Cox proportional hazard model was defined, and it allows a full regression analysis of censored data. Regression, this purple is designed to remind you that this equation looks very much like regression. And if one is a practitioner and using an R package, for example, that's like a regression, very, very similar. One doesn't even know sometimes that what's inside is not regression. So far, wonderful. But the community was worried since long ago with the limiting assumption. And the limiting assumption behind the Cox proportional hazard model is that this part in red, so you see it's a sum. What about higher interactions between the terms? What about nonlinear interactions? Biologists with their neural network organizations made us already think that it's not really a sum, so that is something else. And also, given that there will be a regularization, not many features will enter the final equation. There will be only some. And how about the so-called weak covariates? In the past 2 years, or maybe last year, but very, very recently anyway, there has been new biomarkers defined that are indeed not a compact, stingy sum of important variables, but hundreds of the so-called weak covariates. Covariate, feature, attribute are used interchangeably, meaning the same thing. So that is what comes on the left before the response variable. So it should not be a sum somehow. Again, I remind that we are not talking about pictures. We are not, because then it would be most likely convolutional neural networks. I don't know, but I guess so. And so we're talking about data where P is much greater than N, and the type of attributes are genomic and clinicopathological attributes. There has been plenty, hundreds of articles published from 2017. Oh, yes, 2017, yes, until now. And they all report some progress being better than Cox pH, Cox proportional hazard, and Cox model does not need to be proportional hazard only, but all right. So they say, let's even say that they're better than Cox. And yet there is a mystery. The mystery is that people like myself are very sensitive to new algorithms, because new algorithms give better success rate and new functionalities. So we are very eager to use them in our everyday analysis in order to make our results better, go to higher standard journals. Definitely there's a motivation. And yet from 2017 until last year, there was no single application of all those hundreds of algorithms, at least dozens of new deep learning based algorithms in the clinical analysis of data. That sounded fishy, and I started to investigate. So misconception number one that blows up an attempt to propose a new algorithm and convince a practitioner to use that algorithm is that the C index is of 0.64. The C index is it's an analog of area under curve. So let's say if one is classified cancer patients and non-cancer patients for a serious, expensive, and intrusive manipulation, and they tell you that their area under rock is 0.64, well, I bet you would be very worried. I bet you would not go. So that's almost random. The C index is the ratio where the predicted survival between the two randomly chosen points is correct, divided by the entire number, the sum of those pairs. Oh, it's something like accuracy, but an analog would be in the application. But to be more precise, that's area under rock. And area under rock, when it is completely random, it's diagonal, and it's 0.5. Moreover, in computer science literature, they quite forgot that it can't be below 0.5. And they were saying, well, if that is below 0.5, then one needs to work. And then it's just above 0.5, then just excellent, then just take the best curve. No, no, no. One should be alive to the realities of the application where one works. So misconception number one, even though the proposed algorithm work better than the so-called Cox model, the way the computer scientist views this Cox model, it It still is not enough to give 0.64 because it would be 0.85 would be excellent. So above 0.80 is a practical high enough value for a biomarker to be in moral right to be submitted to a journal, or anyway accepted at a clinical journal. Again, if anyone is in doubt, think about the C index is as an analog of the area under curve. Misconception number 2. Cox pH. First, Cox pH is not the only Cox, is not what Cox proposed a broader model. And what people do, what computer scientists do, is they usually take a very, very vanilla version of a library, while a practitioner works with the best. With the best, I guess, is the Glemonet package in R. And if one searches, one finds the new package, the best, published a couple of years ago. Therefore, the baseline, the state of the art, whatever you call it, is cheated on. I call it cheating on the baseline. And almost invariably, on all those dozens of articles published, almost invariably the very vanilla version was taken. I have been critical about computer science, but isn't that a world of new possibilities? Well, obviously, yes. There have been definitely advantages that the deep brings in. What are those? Biologically aware neural networks. What is that? That is the possibility to take the ontological organization of the genome and simulate it with the neural network. I am not aware, and I have carried out a Google search this September, and I am not August, September, something like this. I am not 2024. I'm not aware of any clinical, real application of it, but it certainly is a wonderful possibility, and most likely a couple of years more, and it may work indeed, and that would be a very potent tool to understand the disease. Right. Secondly, one can criticize deep as long as one wants, yet there have been novel biomarkers designed and published with no statistical counterpart. So indeed, weak covariants have done the job. Indeed, not the classical stingy equation, but several hundreds of genes. And with a neural network, not with the Cox model. (2024) is the first year when those ideas on deep neural networks in survival analysis have appeared in clinical journals in almost simultaneously. Within the difference of days and months, several works reported having used this or that algorithm. There will be quite many by now proposed for survival analysis. All right, by what I've just said is I meant that although deep should be taken very carefully, there still is a need for better tools. And there's always wishful thinking an analyst is left with when results are not so good. If only I had more data, my conclusions would be sounder. There's plenty of very inspiring research in pattern recognition. My excellent, most loved journal is pattern recognition, and it kind of gives hope to my wishful thinking. we come to the last part of my talk, called the artificial intelligent construct, the brain, which is about the following. Typically, an analyst works with a very modest, small data set. And as I've said, one thinks as if it would be 100 patients more than I would be able to prove, and now my p-values are not good enough, and I can't do that, and I couldn't do that. So what is those new tools that give a hope? Pre-training. One has read that leakage here is a big problem and should be taken very, very carefully. But still, there's a road. Maybe there are obstacles on the road, but there's a road, pre-training. And in this diagram you are viewing, I'm not making any specification of what the architecture would be. It would be several architectures tried and obviously automatic tuning of those architectures. There's also a thing is relabeling. Via a diverse, that is possible to achieve via diverse and application-independent distances. For example, it could be with a neural network. If we try to be modern in this talk, if I try to be modern in this talk, so it can be done via CIBEREHD network, for example. Therefore, I have my data, but I also can get more data if I relabel those data points with the use of a distance function. And this cloud, gray cloud, indicates a lot of experience, a lot of data sets accumulated during the years of bioinformatics analysis. And this white data set under analysis is what one is working with. So there's an algorithmic way, and there's data, related data, as in pre-training, not necessarily directly related data, but some knowledge which one can benefit from. Well, here, next slide, is should be taken more of a warning. I've said that, for example, with pre-training, there's a possibility of data leakage, and in cancer research, although pre-training is promising and one can say with some preliminary positive results published, due to the idea that different cancers use the same mechanisms, and it's just then later is more specified into a particular cancer. So one can use the lower levels of the neural network to encode those generic biological processes, and the upper layers can be used to fine-tune. One should be very careful, not only about standard overfitting, but also about data leakage. In Nature Methods this year, I guess this August, if I remember correctly, an article was published with 7 questions about data leakage, and they started saying that many computer science, many computer science solutions, many machine learning, to be more specific, solutions are not correct, because they became victims of data leakage. Data leakage can be viewed as a more general form of standard overfitting. It is defined as an illegal information in some way passed between the attributes about the class value. For example, an analysis, an example of leakage can be when individual features are correlated with the response variable, and in fact what the model has learned is individual features. The model in fact learns to recognize a patient rather than recognize a disease. And yes, we have a current article on that, on those 7 problems and more under review. We have programmed several heuristics against the leakage. The way the leakage is understood currently with those 7 questions, for example, because the authors of who write on information leakage always emphasize that this is the current understanding, that a little bit more, and will capture more problems and will understand them and will add to the definition. Once there's an understanding on what is a problem, one can computationally devise a procedure that finds those problems automatically, hopefully, but not necessarily. So we are trying to pursue this line. We are trying and indeed programming heuristics against leakage. A trivial example of leakage, so trivial that it often is not even considered as leakage, but which I unfortunately quite often see in published articles, is that data sets are normalized. The data set is normalized at in one step, to put, for example, the whole distribution to have mean 0 and standard deviation 1, and then this dataset, global dataset, is divided between train and test. But that is not such a big problem, because central journals, for example, Bioinformatics, have a checklist for quality. Therefore, if one is aware of that checklist and is careful, if the journal makes one to check, to explicitly tick that, yes, I've checked that, or one's libraries are throwing an exception if one, or a warning, throw in a warning if they have detected a problem of this type or another one. That is quite manageable, and yet there are quite a lot of errors still in practical analysis, unfortunately. Questions can be sent to my emails. I have painted half in green, half in purple, being back to my metaphor that one is green and the dashes and sleep furiously or achieve furiously, and the other part is thinking, all right, achievements and promises are very, very good, but there are plenty of responsibility and the cost is high of an error. Recent articles on the topics I have just talked about are a review on deep learning survival for omics and clinicopathological data, where we concentrate on most important architectures and the issues we find critical on the way of those algorithms coming into practice this year, 2024. Indeed, those ideas have found their implementation in clinical practice after, 2017 to 2024, that would be 7 years of research progress, with a very intelligent, I would say, article by Katzman, Deep Survival, intelligent in the sense that they were realistic about the challenges, and they charted the math of further research and consequent dozens of articles developing that road that finally, yes, have come into practice, that finally, yes, a very good alternative to the gold standard in clinical statistics. Another article I would like to mention here, published the previous year, 2023, is a commentary in a journal where we brought up two problems. That first is, please do not forget the physical, I would say physical, material, tangible meaning of what is a C index, of what is a success metric, and how it should be interpreted. This is what I've said, that if one publishes a biomarker, the C value has to be 0.8, not 0.64. That is obviously, very obviously not enough. And the second thing was there about calibration, so some other aspect of what adds to the C value. It's not only one C value, it also is about clinical utility, but that is another story. And if organizers will invite me next year, maybe I will talk about clinical utility indeed. Thank you very much for your attention. Let me give you a special issue invitation to an MDP, International Journal of Molecular Sciences, first quartile of Web of Science, where I serve as one of the guest editors. The issue is devoted to exploring cancer molecular mechanisms with artificial intelligence, and not to offend, without an intention to offend the computer science public. You see, I've put it in purple. So it means algorithms, no, are not welcome. What is welcome is solid, new knowledge with inferential proofs concerning new molecular mechanisms for cancer. Articles would be published on the ongoing basis, and the deadline is end of this year. Thank you very much. As a metaphor and the conclusion of my talk, there's this small Sancho Panza and wonderful Don Quixote. And it is still in one researcher. So let's hope we achieve. Hopefully, see you next year. Thank you very much again. And questions are very welcome.
Can I ask a question?
Yeah, sure.
I'm working in AI related to brain diseases, cognitive science, and AI in a near future. Now, at my talk at 4:30 IST is on AI robots, which would be simulating clinical psychologists and the psychiatrists in the near future, going in COVID areas and the rural areas, as well as Hiroshima, Nagasaki. And we have already done the work in that direction, NLP chatbot, which I will discuss today. So your work is on AI. So can there be some sort of I can write to you for some collaboration if you are doing some projects or something related to it, we can further discuss it?
Yes, absolutely. I would mostly welcome any sort of collaborations, especially in neuroscience, because we work on neuroscience. We work even on psychiatry with some recent publication a year ago. Yes, sure. There's
okay. Then I'm Dr. Shweta Gupta, and I am a senior member of Hong Kong Group of Conferences, very well renowned in China for my work and in Japan. And I will certainly write to you. I'm from India, Dr. Shweta Gupta. I will write to you about my work. I am basically an AI engineer.
You know what we have done? We have some years ago, again, for a psychiatric, rather neurological disorder, we built a machine which mimics the human ear. So we proved that the machine works in the same way that a group of people would perceive the speech of a diseased person. So we try to, you see there are at least two types of computation. The computation that happens in the brain, and which we as humans, how we assess other people as being sick or not being sick, as being communicationally adequate or not. And also there's another type of computation, computer. So that is magnetic 0 and 1. And we prove that what the computer does is what the conclusions of the computer would simulate the conclusions of living beings, of persons.
Yeah, and I had recently done work on depression. A depressed face will, through computer vision, the face recognition will be done, and through the biomarkers like the chapped lips as well as frowned eyebrows, it will and the speech recognition, the consistency in the speech, etc., will be monitored to find that the person is in depression or not, or to which extent. And further, the NLP chatbot would help the person in anxiety, and it would be a human kind of touch in the chatbot, which I will be discussing today at 4:30 IST. So that comes from the NLP point of view. So this kind of work I have done till now with my
That's
students.
mostly exciting and interesting indeed.
Okay, thank you. Then I will certainly write back to you for any other collaboration.
Looking forward to receiving a communication.
Thank you so much.